The next section of Costanzo's respiratory physiology chapter discusses how carbon dioxide is transported in the blood. There are three main forms of CO2 carried in the blood. Some of it's dissolved, just like the dissolved oxygen we talked about. Some of it is carbaminohemoglobin, carbon dioxide bound to hemoglobin, and bicarbonate is a chemically modified form of CO2, and quantitatively the most important form. Just like with oxygen, a portion of the carbon dioxide is carried as dissolved. We can use Henry's law just as we did for oxygen, stating that the concentration of CO2 in the blood is the partial pressure multiplied by the solubility. The solubility of carbon dioxide is 0.07 milliliters per 100 milliliters of blood for every millimeter of mercury. So, the concentration of dissolved CO2 in arterial blood is calculated by Henry's law as 2.8 milliliters of CO2 per 100 milliliters of blood. 40 times 0.07. This is approximately 5% of the total CO2 content of the blood. Remember, there's a lower solubility for oxygen compared to CO2. So the dissolved O2 is only 2% of the total oxygen content of blood. Carbaminohemoglobin. CO2 binds to the terminal amino acids on proteins, for example hemoglobin, and other plasma proteins such as albumin. When CO2 is bound to hemoglobin, it's called carbaminohemoglobin, and it accounts for about 3% of the total CO2. CO2 binding to hemoglobin occurs at a different site than oxygen binds to hemoglobin. As we talked about previously, carbon dioxide binding to hemoglobin reduces the affinity for oxygen, causes a right shift of the oxygen-hemoglobin dissociation curve. This is the Bohr effect. If we have tissue that's exercising, producing more CO2, it right shifts the oxygen-hemoglobin dissociation curve. Also, oxygen bound to hemoglobin changes hemoglobin's affinity for carbon dioxide, so that when less oxygen is bound, the affinity for hemoglobin for carbon dioxide increases. This is referred to as the Haldane effect. The mutual effects of oxygen and carbon dioxide on each other's binding to hemoglobin make sense. In the tissues, CO2 is produced, binds to hemoglobin. Hemoglobin's for affinity for oxygen is decreased. Therefore, it releases oxygen to the tissues more readily. In turn, the release of oxygen from hemoglobin increases the affinity for CO2 that is being produced in the tissues. Almost all the carbon dioxide that's carried in blood is carried as a chemically modified form, bicarbonate. It accounts for more than 90% of the total carbon dioxide. The reaction that produces bicarb from CO2 involves the combination of carbon dioxide and water to form the weak acid H2CO3. This reaction is catalyzed by the enzyme carbonic anhydrase, present in most cells. H2CO3 dissociates into hydrogen ions and bicarb. Both of these reactions are reversible, and carbonic anhydrase actually catalyzes both the hydration of CO2 and the dehydration of H2CO3. So that CO2 plus H2O goes to H2CO3, which goes to hydrogen ions and bicarbonate anions. In the tissues, carbon dioxide generated from aerobic metabolism gets added to the systemic capillary blood, converts bicarb by the reaction described previously, and it's transported to the lungs. In the lungs, bicarb is reconverted to CO2 and then exhaled. Figure 524, which is on page 216 in the text, describes this. In the peripheral tissues, metabolism produces CO2. The CO2 goes into the capillary, into the red blood cell, where carbonic anhydrase facilitates the conversion of CO2 into bicarbonate. If we go through figure 524, the circled numbers correspond to the following steps. Let's take this systematically. Number 1. In the tissues, CO2 is produced. CO2 diffuses across the red cell membrane. The transport of CO2 across these membranes occurs by simple diffusion and is facilitated by the large partial pressure gradient for CO2. Step 2, carbonic anhydrase, found in high concentrations in red blood cell, catalyzes the hydration of CO2 to form H2CO3. Then in red blood cells, the reaction are driven to the right by mass action. CO2 is constantly being supplied from the tissues, forcing the equilibrium of this equation to the right towards bicarb formation. Step 3. H2CO3 dissociates into hydrogen ions and bicarb anions. The H plus remains in the red blood cells where it's buffered by deoxyhemoglobin, and the bicarb is transported into the plasma in exchange for chloride. For step 4, the hydrogen ions produced in these tissues remain free in solution. If they did, they'd acidify the red blood cells in the venous blood. So we got to buffer the hydrogen ions so that the pH of the red blood cells and the blood remain within the physiologic range. The hydrogen ions are buffered by the red blood cells by deoxyhemoglobin, and it's carried in the venous blood in that form. It's interesting that deoxyhemoglobin is a better buffer for hydrogen ions than oxyhemoglobins, but it makes sense because the venous blood is more likely to have higher hydrogen ion concentrations. By the time blood reaches the venous end of the capillaries, hemoglobin is conveniently deoxygenated. It's around to carry the hydrogen ions. This useful reciprocal relationship between the buffering of hydrogen ions and deoxyhemoglobin is a further extension of the Bohr effect that we discussed. It states that an increasing hydrogen ion concentration causes a right shift in the oxygen-hemoglobin dissociation curve, causing hemoglobin to unload its oxygen more readily to the tissues, conveniently allowing it to carry more hydrogen ions. So the hydrogen ions generated from the tissue, CO2, causes the hemoglobin to release oxygen, and the deoxyhemoglobin that's released its oxygen can be a better buffer for the hydrogen ions. Moving on to step 5, the bicarb produced from these reactions is exchanged for chloride across the red blood cell membrane to maintain a charge balance. The bicarb is then carried to the lungs in the plasma of venous blood. The chloride-bicarb exchange, or the chloride shift, is accomplished by an anion exchange protein called band 3 protein. It's called this because of a prominence in an electrophoretic profile within the blood. Then, all of these reactions that occur happen in reverse in the lungs. It's not shown in figure 524. Hydrogen ion is released from its buffering sites on deoxyhemoglobin. Bicarb enters back into the red blood cell in exchange for chloride ions. Hydrogen and bicarb combine to form H2CO3, which dissociates into CO2 and H2O, and the regenerated CO2 and H2O can be exhaled in the lungs. That's our discussion of the transport of carbon.